Sawadika. Hi. Thank you for your nice introduction and welcome you all to Bangkok. So today, okay, thank you. Today, I'm going to talk about the application of long-read sequencing, not a whole genome sequencing, but it's target gene sequencing. It's just the HLA sequence. And we did that on the patients who are having PRCA associated with the use of erythropoietin or EPO. So I'm going to divide my talk into three parts. Give you some background about PRCA and a little bit of long-read sequencing of HLA and the problem with the current technology, and I'll show you some relationships that we found. So first, has anyone in this room ever had PRCA before? Hopefully not. Well, I've never ever heard about this before until I came back from my training, and we saw some patients who having anemia after prescribed with EPO. Those patients who have severe red cell anemia, just only the red cell, and it's marked reduction on the absence of erythroid precursor from the bone marrow. Well, actually, PRCA is a condition that can be caused by several etiologies. It can be congenital, it can be primary acquired, and it can be from several secondary causes, and drug and toxin are one of them. Several drugs are linked to PRCA, and recombinant EPO is one of them. People with CKD, especially advanced stage of CKD, will generally be anemic by disease itself, and they require treatment with erythropoietin for uplifting, for stimulating the bone marrow to produce more red cell. But some patients who are unluckily having some resistance to EPO, and in the other term, they'll be more anemic from having EPO. In Thailand, we started having EPO in 1997, and after that, the case with PRCA is getting higher and higher, and it's becoming a very important problem in nephrology. And I'm sorry this one in Thai, but it's the report from the Ministry of Health, Thai government, saying that, well, during the year 1998 to 2001, the case with PRCA in the world is getting lower and lower because of the better way of dealing with all the EPO they used. But in Thailand, the incident is still continue getting higher and higher every year. The CKD case in Thailand is about 1 million people. 16% of them would be in CKD stage 4 and 5. So that means it's about 150,000 or 180,000 patients estimated to be on EPO each year. And the incidence that we had in Chulalongkorn University is about 200 to 300 PRCA cases per year, which is underestimate. PRCA is very region specific, and it happened in Thailand like 1 to 600 patients, but it's very low incidence in the US and other part of the world. The early first case that happened in Chulalongkorn Hospital was reported back to 2005 by Professor Kiet group. They found a patient who required a lot of packed cell treatment after having EPO, and they continue having anemic until they have kidney transplantation, and they get survive because they stop using EPO. After that, they continue having series of cases with this disease for several years. There are several factors that has been validated to be associated with PRCA. The first one is the route of administration, changing from subcutaneous to IV would be better. Then the cold chain, the way to keep EPO. And if the patient can tolerate EPO, they can tolerate longer, that is better. Or the product quality, because EPO in Thailand is usually a biosimilar product or a biocopy. The innovator product is very expensive, and we cannot really afford it. All these things has been found to be a factor associated with PRCA, apart from genetics. Okay, the genetic that we think that is have a very high role in this thing is HLA. HLA is part of pharmacogenomics, which is the study of people's genetics to affect body response to drugs. And because everyone has taken drug at some point, so drug that given to people can lead to the desired response, or some people who have a modifying genetics may have moderate response to that drug, so they need no dose adjustment, or unluckily some people will happen to be on toxicity, so that drug has to be avoided. And I really like this figure from Professor Sir Munir Muhammad. He summarized the important application of pharmacogenomics into 6 different aspects. The first one is to predict drug response, improve efficacy, and preventing ADR, which HLA is one of this part, and then predicting the activation of the product, develop target for gene therapy, and has a role in drug discovery. The HLA is on chromosome 6, and it has 3 classes, class 1, 2, 3. Class 1 and 2 is very important in the daily immunogenicity. And if you go to CPIC website, which is the website that summarizes all the information about pharmacogenomics, and I did this table by myself, you can see, and you cannot read because it's too small, that there's a lot of report linking HLA to several drugs. And I summarize this fuzzy little information into this figure. So HLA has been reported to associate with toxicity like scar, DRESS, DILI, or granulocytosis. There's nothing related to PRCA. But we think, and we found, that HLA-DRB109 and DQB103 are linked to the toxicity called PRCA associated with EPO. The first study came out in 2009 with our group, having only 22 patients. They had the HLA genotype by, as far as I remember, is PCR based. And all of them has 09, DRB109, and some has DQB103. And the odds ratio is about 2.89. After that, we have a better technology for HLA sequencing came out. Well, this is the figure to summarize the, I don't know if I had, yeah, the HLA typing methods. You can go from serology technology. That was my mentor's time, Professor Warasa. 1960, right? And then molecular assay, that's my medical student time when I first met him in the medical school. And then move on to here, the 3rd generation sequencing, long-read sequencing. The problem about the hybridization-based method, which is currently used today in Chulalongkorn hospital for doing HLA genotype, is Luminex. It can give only the 4-digit typing, HLA, for example, 0201. That's it, right? But for long read, it will give you the full length HLA, and it will give you up to 8 digits of HLA. The problem about 4 digits typing, for example, carbamazepine here, we know that carbamazepine is associated with HLA-B 1502, but we found that within this group, it has two different types, O2 0101 or O2 0104. And we don't really know which one is closer linked to the toxicity from carbamazepine. Okay, and you all know that long-read sequencing is a powerful tool in every field, including pharmacogenetics. And we did a further study with long-read sequencing technology with PacBio Sequel 2E, having 157 case and 56 control. We trying to match them in every aspect, including the EPO type that they had. And we do the long-read sequencing and analyze for HLA A, B, C, DRB1, 3, 4, 5, and DQB1 and DPB1. We used the GenDx software to analyze the HLA from this from the sequencing file that we got. And we found, I'm sorry, this one is quite tiny. We found 30, within this about 200 cases, we found 34 types of HLA-A and more on B and C, DRB1, DQB1, and DPB1. You can see in orange is case, in blue is control. There are some differences, right? So we do the multivariate analysis with the covariates that might influence PRCA, including the type of EPO that they received. And we found that HLA-B46 and DRB109, which is the same thing that we found on 2009, highly linked to the risk of PRCA in this group of patients. And we also noticed that B46, DRB109, and DQB103 are linked. They are linked together as a haplotype. And we also did some further work showing that number B46 and DRB109 are highly bind with EPO with better, higher affinity, far from other HLA. When we compare our data to some other studies in the world, there are only 4 studies in the world because, as you can see from my slide, that the incidence in the US is very low. The Chinese also report some association of DRB1. Europe and Canada, they also report DRB109 with a very, very high odd ratio because their case is very low. And our study with this one, we found that 46 associated with the risk with odd ratio of about 5, and DRB109 is about, the odd ratio is about 3.7. So now we are quite certain that we can put these 2 types of HLA into this figure, especially for Asian country. Okay, so I will finish my talk with this slide. For take-home message, Thai CKD patients receiving EPO are at risk of PRCA. And the risk is small, which HLA B46 and DRB109. The long-read sequencing is the platform for HLA sequencing. And Nina mentioned that it's very important to have long-read sequencing in kidney transplant patient. I didn't know that she would mention this, otherwise I would change my talk, because we already have a long-read sequencing for 800 patients having kidney transplant in Thailand. And so next time, if you invite me, I will give you, this presentation. Okay, so finally, I would like to thank my team. I have my mentor, Professor Varasak, here, and some of my colleagues, and also the work are finished by my PhD students and also the ladies here in my lab. Thank you very much.
Thank you, Professor Pajaree. Thank you for sharing wonderful research results. to